So, good morning, welcome everyone, and thanks for joining us today. And welcome also to those who are online. This meeting is made of three main parts: a short introduction of the project and of the team, two guest lectures, and finally a space for questions and discussions. So for those of you who are online, and in any case you have any question, please write your name in the chat so we can keep track of the order. We wish to record the event, so if any of you is against this, please let us know immediately. Otherwise, we proceed with the recording. I thank Nish, who we are in a beautiful, a great building in the center of Venice. Nish is the new Institute Center for Environmental Humanities that is affiliated to the project and is hosting us today. So I thank Nish, and this project is based at the Department of Philosophy and Cultural Heritage of the University Ca' Foscari of Venice. So HealthXCross is a starting grant project funded by the European Research Council. It has started in September 2021. It will last 5 years, so ending in August 2026. The project is a multi-sided ethnographic and comparative study of how microbiome research is reconfiguring practices and ideas of health. It has been conceived as an anthropological project, but is in dialogue with many other disciplines. Microbes and the microbiome. Can we share the screen? Microbes and, sorry, arrivare alla seconda slide. Microbes and the microbiome have in recent years attracted lots of interest, both in the public, but also in the social science and the humanities. This currently debate have explored how we live with microbes and how microbes can renew our social imagination, concepts, and practices. Microbes indeed are connectors that radically change our ideas of what a body is, because microbes entangle bodies and environments. This project has been inspired by these debates, but it also takes a step back and asks, what are we really referring to when we speak about the microbiome? We do not take the microbiome as a given. We do not consider it as a stable scientific object. Rather, we question how it emerged as such. And given that the same concept of the microbiome is, first of all, a technological revolution, we look at the microbiome as a part of a data-driven science that is creating new knowledge at the intersection of landscapes, labscapes, and technoscapes. So we will exploring how this kind of research, microbiome research, is changing biology and biomedicine, but at the same time, and in turn, is also changing anthropology, the social sciences, and the same experience of being human. The project has three main research clusters. So, three main research clusters. One is diversity, that looks at how scientists by aggregating microbial samples deriving from very different origins, so humans, non-human, soil, plants, air, whatever, we make notions of biological diversity that cross the conventional categorization of space-time and food. The second cluster is called space-time of innovation and looks at how innovation in microbiome research emerged from collaboration between the global north and global south. So those are issues related to local regimes of knowledge, but also to the transnational governance of open data. The third cluster is health, and it explores how microbiome research shapes new trends in healthcare and health governance. So we connect here to debates about one and planetary health. We have six case studies, two in Europe, two in the United States, one in Africa, one in the Pacific, and one case study in Asia will add thanks to a recent funding from the Italian Minister of Research, the framework FARE. So, but instead of continuing bothering you with my voice, I let you entertain with a short video, a short animated video that illustrates the main research questions on the project and its theme. It's not starting. Please bother with us as people interested in science. You know that science, that technology needs some negotiation. So.
Bueno.
No, non si vede. Sì. Ancora una. Prossimo slide. Non mi va, non mi va. Se no ci metto online. Possiamo andare direttamente online.
Microbes are tiny living things that are found within, on, and around us, and are too small to be seen by the naked eye. The most common types are bacteria, viruses, and fungi. The human body is home to millions of these microbes, but they also live in animals, plants, water, soil, and in the air. Microbes are everywhere. Microbes are the connectors between us and the environment in which we live. Health is a concept usually associated with humans, but how can we rethink human health as part of an interconnected environment? HealthXCross will explore the implications of approaching the planet as a body and the body as a planet. We are a team of anthropologists exploring laboratories where scientists analyse microbial data collected all around the world, derived from different samples, crossing space, time, and species. This huge amount of data is then aggregated into a supercomputer in order to develop scenarios of health for both us humans and the environment. Anthropologists and scientists will collaborate to understand what it means to be human and to live on an interdependent planet.
Sí. So, this is the plan, but of course, being an ethnographical project, then the project will take its own life. And it will be an adventure, and the researcher that will give the real protagonist that will give life to this provisional plan are 5 researchers so far. Maybe others will add toward along the life of the project. And today these researchers will introduce themselves. All of them will start to work in November this year, except one, Lucilla, who has already started to work with me last November. They are all very skilled researchers with varied backgrounds and experiences, and I'm very excited to work with them and with them to establish a collaborative framework also in the social sciences. So I let the floor to them, please. So the first one is Lucilla.
Good morning to everyone. So as the first postdoctoral fellow of the project, it's an honor to have the opportunity to address such a distinguished audience and to briefly introduce my contribution to HealthXCross. So over the course of the year, I have been conducting an ethnography of a data collaborative of interdisciplinary scientists set up online at the beginning of the COVID-19 outbreak. To examine a wide range of health issues at the human, animal, and environmental interface. Data collaborative are networks of stakeholders and institutions that meet to gather and exchange data and try to solve social problems or try to put a vision, a framework into practice. And the data collaborative that I've been studying was committed in examining a wide range of health issues, as I said, by integrating, collecting, sharing, and analyzing data from different data sources and analysis. So the framework that guides these data sharing efforts is One Health. One Health is an approach that aims at sustainably balance the health of humans, animals, and ecosystems. So it has a transformational promise. It contains a visionary life work that these scientists are trying to put into practice. And from their perspective, the use of big data technologies such as big data can help from their perspective to achieve this One Health promise. So I've been studying and following their activities and data sharing efforts, and it appears that the practice of digital interdisciplinary collaboration is unstable. It builds on precarious scientific collaborative efforts, uneven technological infrastructures, and a global data governance landscape that is still information. So the point of my ethnography is that we need to look at the digital infrastructure of collaboration in which One Health as a vision operates and become transformed into a research project. And to conclude, the chances to achieve the One Health vision depends on how we are able to rethink the subjectivity of data and data work and the diversity of epistemic spaces of collaboration that we build as scientists to address societal and environmental challenges in a time of global collapse and data-driven science. I'm very excited to bring forward this research this year, and a very warm welcome to the HealthXCross new member teams, and thank you, Roberta.
Hello to everybody. Can you hear me? Maybe switch that. Okay, now it's better. Good morning to everybody. I'm Valeria Burgio, and thanks for inviting me here. I'm very excited to begin this new experience with this interdisciplinary and international group. And I will tell you just a couple of words about my background, because I'm a little bit at the side of anthropology. And I'm actually, I have a degree in communication science and a PhD in art theories. And my PhD thesis was about the language of animation, and in particular on the artist William Kentridge from South Africa. But then I moved to design, to teach and research in design schools. And I taught theory of visual communication and visual culture for some years. And my last research field was on infographics and data visualization. And it was the moment in which I began to study how apparently neutral and objective images were actually constructing knowledge. So this was the bridge towards this project, in which I will mainly deal with microscopical photography and how those kind of images are actually constructed and dealt upon and discussed. And so they are a result of a dialogue. And so I'm very happy about having a brand new corpus of images that I will have to study and compare. And also, thanks to the ethnographers that will work in this project, also recording and transcriptions of the discourse upon those images. So it will be, my part will be in connecting images with the discourse on images. Another important element of my research will be about the circulation of image. So what happens after the images get out of the laboratory and go the public arena. And in particular, I will deal with the artistic practice on microbiome and how this new forms and research of expression add something to the microbiome. Okay, the images of microbiomes and the microbes. Okay, hope to have been clear. And thanks to all for inviting me, and I leave the word to the next researcher.
Hi, good morning. Good morning, everyone. My name is Victor Secco, and I am currently a PhD candidate in social anthropology at the Department of Social Anthropology in University of Manchester, and where I have been following researchers ethnographically, microbiologists and Hindu priests who have been working with the waters of the Ganges River in North India. And more specifically, the scientists I've worked with are interested in bacteriophages, which are these viruses that infect bacteria and then it can be used and are promising in the use of treatment of antibiotic-resistant infections in humans. So these viruses are abundant in the Ganges, mainly because of the influx of sewage and the high number of people that come in contact with the waters because of ritual purposes. So my PhD thesis then explores how these waters of the Ganges River are mediated by different kinds of knowledge practices in terms of microbes, pollution, or religious rituals. And then they're connecting these different scales of humans, non-human, and environmental health concerns through water. And so I'm very excited to join the team of HealthXCross and explore further how technoscientific efforts to understand microbial worlds reconfigure ideas of health, bodies, and environments. At HealthXCross, I'll be continuing working between laboratories and landscapes, and I'll be joining bioscientists in Europe at a laboratory in Europe that are working, trying to link the micro and the macro scales through data collecting across time, space, and species. I hope to explore ethnographically these concepts, the tools, and the data that are being created in the current microbiological knowledge, and with my colleagues at the HealthXCross, and also with the bioscientists advancing this important work of linking together, building bridges between social and biosciences as well. So I'm very excited to be here. Thank you very much. And it's good.
Hello, everyone. Can you hear me okay? Hello, my name is Joanna Pomosini, and I'm an interdisciplinary researcher of the life sciences. I work ethnographically across science and technology studies and history and philosophy of science. So I do a combination of ethnographic observation and conceptual analysis. And my PhD, which I'm currently finishing at the University of Copenhagen's Medical Museum, is on how microbiome research as a field across fields of discourse is changing representations of the human body in the relationship between body and environment. And so I'm very interested in a renewed human figure through microbiome research, which includes aspects of the environment. And in that figure is the key to my contribution in this project, which is in the health cluster. And of course, the human body that we treat medically really matters. And so I'm very interested in how health intersects with sustainability through microbiome research, and that's what I'll be bringing to this project. I'm so excited to be here with this fantastic team, and looking forward to what unfolds. Thank you.
Hello, everybody. Can you hear me? Thank you for inviting me to the floor, and I'm very excited to be here and to explain a little bit about my career and also my possible contributions to the project, to the HealthXCross project. I am currently a postdoc at the University of Milan, but I completed a PhD in Germany, in Bayreuth, in anthropology and African studies. So my field of expertise is African studies, where I specialize geographically in North Africa. And I completed a PhD within the frame and with the funds of an ERC working on slavery. So I was working with race and racism in the meaning of racial discrimination because of like dark or black skin tone, but also as the legacy of specific historical phenomena, such as the trans-Saharan slave trade in this case. But also I was working on issues of identity, ethnicity, and race, which are like, in this case, are very connected vertically to genealogy and ancestry and descent. So working with these things means also working with post-colonial lenses, like studying how ideas and practices are mobilized and moves across boundaries within the continent, so the African continent, but also between like global North and global South. So that's what I wish to bring in the project as a perspective, because I wish to work with the research trend of HealthXCross, which is working on the analysis of the effects of hierarchies that hierarchies have on the diffusion and the movement of science, of technology of science, and of knowledge, so how it moves across borders. So my fieldwork is not yet defined, but we identified like some possible contributions, some possible research trends. One is the question of intellectual property and data collection in the continent, and how they also move within like directions, global North and global South, but also across national borders, like within the continent, the African continent. The second would be the effects that these hierarchies have on the perception of science within the continent, meaning how local ideas of science are shaped, how they overlap and contradict, and how they intertwine with Western ideas of science. And three, there is a very emerging and engaging debate nowadays in Africa about identity politics and the connection between identity politics and Western modern scientific tools like DNA tests, for example. So there is a huge debate on what is Africanness and what is African identity, also through the employment of scientific tools such as DNA tests and these things. And from our preliminary contacts with some research labs, we also found out that microbiome is part and is involved in the process of defining and shaping what Africanness is in some places. So these are my possible contributions, but I'm very open to recommendations and suggestions. And I thank you for your attention.
So, thank you very much. Now we enter into the second part of this event that will be two guest lectures. You know, I need to really make a premise, because this project is, I mean, I'm here speaking, but I'm here now, but it's not just me, you know. This project is the result of many encounters I had in my academic life, many intellectually generous and smart colleagues who nurtured me, who supported me in many ways. So in writing this project, I was inspired and supported by them, and I owe to them a lot. Today here, there are two of them, two exceptional scholars who generously accepted to offer a theoretical commentary to the project. One is Hannah Landecker, who has been key in helping me to develop the project and still is a great source of inspiration. The second speaker is Jamie Lorimer, who has recently published a book entitled The Probiotic Planet, Using Life to Manage Life, which closely speak to the issue, to the concerns of this project. Therefore, I'm particularly glad and honored to have them both today. So the first speaker is Jamie Lorimer. Jamie Lorimer is Professor of Environmental Geography at Oxford University. His research interests encompass cultural geography, the geography of science, the politics of nature, and wildlife conservation. As already mentioned, he's the author of the book, The Probiotic Planet, and also of another monograph entitled Wildlife in the Anthropocene, Conservation After Nature. So, Jamie, do you hear us? Are you there?
Thanks, Roberta. Yes, I'm here. Can you hear me okay at your end there?
We hear you. So thank you very much for being among us today. And I let you the floor.
Brilliant. Well, thank you very much for having me and for your kind words. And I'm sorry not to be there with you in Venice. I'm going to share my screen. I have a PowerPoint presentation, which will, I need the host to enable PowerPoint screen sharing, please, on the Zoom settings.
Privacy, vero? Sicurezza, gli devo permettere di condividere.
Devi andare sul utente.
Già fatto.
Okay.
Okay, hopefully you can see that now. Is that clear? Yeah, brilliant. Okay. Well, as I say, thank you very much for hosting me today virtually. And I wanted really just to say a few words to set the scene about the current microbial moment, if you like, to try and think about why we are gathered here, perhaps why the ERC has funded this project on the microbiome, and why social scientists are interested in the microbiome, why, if you like, there's a new alliance emerging here between science and the social sciences at the contemporary moment. So the three aims of the talk today are to provide a little bit of a background to the rise of the microbiome. There's obviously an awful lot that could be said there, but to flag some contemporary dimensions that I think are pertinent for understanding why microbes, why now. And then towards the end to raise some critical questions for the project, many of which are very explicit within the outline of the project that I've been able to encounter, that might guide some of the research that is going to be undertaken over the next 5 or 6 years. Okay, so the background to the rise of the microbiome may well be familiar to most of the people in the room, but just to provide some context. So microbiome science emerges largely out of the Human Genome Project and the massive cheapening in the costs of sequencing that happened as a result of the investment into next generation sequencing that happened through the early 2000s. And that technology was deployed from looking at the core human genome to begin to map the life in, on, and around the human body. So the Human Microbiome Project, which completed nearly 10 years ago, established a set of reference databases for looking at the composition of the human, and that work continues. And again, the cheapening of the sequencing technology has allowed microbiologists to expand the scope of microbiome research beyond the human out into a range of projects that are now, with quite grand ambitions, thinking about the Earth microbiome, thinking about the microbiome of the built environment, particularly of buildings and hospitals and other institutions, and then a range of citizen science-led microbiome projects that are looking at things like the microbiome of the cat or the microbiome of the dog. And so there is this proliferation of microbiome initiatives and a great proliferation of data coming online to begin to map and describe both the microbes that compose the world, but also the interactions between microbes, the functions that microbes perform that are coming to be understood as so core for thinking about health. Scientific interest in the microbiome traveled widely and quickly out from the mainstream high-profile journals like Nature into popular publications like Scientific American. It's championed very well by Ed Yong, the popular science writer, in his book, I Contain Multitudes. And you know when a scientific movement is gone mainstream when it features on the front cover of The Economist. So here is The Economist's take on the microbiome. This was in 2012. Microbes maketh man. It's there in the New York Times. It's everywhere within a certain set of publications that deal with taking science into the mainstream. Likewise, it's come to feature prominently in exhibitions in prominent natural history museums. The American Museum of Natural History ran an exhibition on the microbiome in 2015. The Eden Project in the UK has its Invisible You exhibition on the microbiome. And Amsterdam has gone one further and now has the world's first dedicated museum for microbes, Micropia, which is a very well-resourced building just next to Artis Zoo in Amsterdam. Discussions of the microbiome inevitably have come to focus on questions of health and really come to refigure how we think about the role of microbes in health. And this emerging understanding that health is indexed to both microbial absence as much as it is to microbial presence. A growing concern that it is the absence of particular old friend microbes as a consequence of modern forms of hygiene and healthcare, particularly the use of antibiotics, that might in some ways be indexed to the rise of a whole range of autoimmune, allergic, inflammatory diseases. So captured in the titles of these two books here, one by Martin Blazer, Missing Microbes, How the Overuse of Antibiotics is Fueling Our Modern Plagues, and then by Moises Velasquez-Manoff, who's a science journalist, talking about an epidemic of absence, looking at the epidemiological evidence emerging that would suggest that some modern diseases, contemporary modern diseases, are related to dysbiosis, to a dysfunctional composition of the human microbiome. As a consequence, there's been a great rise in investment, biocapital, biotechnology investment, in the words of Janssen here, in mining the microbiome for transformative products. A kind of new resource frontier is opened up as a consequence of this proliferation of data, in which it might be possible to develop various strategies for personalizing medicine through data mining to design interventions into drugs that will transform human health. And then on the more consumer-facing side of things, an amplification of the existing market in probiotics, moving into products both for human diet, but also into products for the human skin. And then this growing interest in fecal microbiota transplant, the ways in which you might be able to create exchange systems for healthy stool to tackle various forms of gut diseases, as well as trying to replicate that in pill form. So we get this sociologically interesting proliferation of microbial knowledge, a growing political and economic interest in the microbiome in Western society at large. And this is mirrored in a growing interest in microbiome science in social theory. Particularly prominent social theorists are beginning to form a new alliance with microbiome science as part of a wider alliance with science that's emerging in various strands of new materialist thinking in the context of concerns about climate change and the Anthropocene. So just to flag some examples that will be familiar, so perhaps the most high profile would be Donna Haraway's latest book, which is based on a close reading of microbiome science and ecological evolutionary development biology, particularly the work of Lynn Margulis, thinking about how our relations with microbes might help us reset our relationships with other people, our relationships with non-human life. Haraway's colleague in the UC system, Anna Singh, anthropologist, has written this wonderful book about fungi, particularly focusing on the Matsutake mushroom, and again, using human fungal relations to think carefully about life in the Anthropocene and the ways in which we might set social relations and economic relations in which a subset of fungi become metaphors for thinking about the social, for thinking about ethics, for thinking about political economy. Other examples would be Myra Hurd's ethnography of Lynn Margulis's life, in which she uses Margulis's theory of symbiogenesis, again, to think about what microbes tell us about society, about ethics, and about evolution. And just a couple of other examples there as part of this wider microbial turn within Western social theory, in which there is an alliance with microbial science emerging, both metaphorically, but also politically, in terms of how social theorists are making new epistemic relations. Okay. So why might this be? Why, if we are interested in the history of science, if we're interested in making sense of contemporary trends within the biological sciences and within the life sciences, why might it be that we have come to be so concerned with microbiome science at the contemporary moment? And there's three themes I think it's worth being aware of here. So the first of which is the ways in which microbiome science offers a new ontology for thinking about social relations, for thinking about human, non-human relations. And in particular, how it departs from the version of the social and the version of the human that was given to us by the modern synthesis of neo-Darwinian biology, which had this great emphasis on individuals and competition between individuals, and particularly premised on the idea of the survival of the fittest and a celebration of competitive relations. So instead, from microbiome science, we get this figure of what Stefan Helmreich describes as Homo microbes, this ontology of the human as composed of microbes. The human is a holobiont, is the concept that we get from contemporary biology, nicely captured here in this advert for Micropia, in which the human here is made up of these sort of blobs that are held to symbolize microbes. Alongside that is a sense that not only is the human a multi-species achievement, the human that's made of many other forms of life, but it's also a human that is fundamentally shaped by the actions of microbes in the gut. So this emerging interest in what's called the gut-brain axis, the ways in which the gut and the microbial activities of the gut might come to shape human mood and human behavior. In extreme forms, a sense that the human is merely a warm, fleshy vehicle for the furtherance of microbial projects, a folding of the human into the non-human, a decentering of the human through a story about, in some ways, the brain evolving as a tool to enable the gut to get about, rather than the gut being in service of the brain, let's say. So a radical decentering of the kind of Cartesian idea of the brain as being central and above the body. And then the third dimension to this is the ways in which this story of Homo microbes folds the human into a wider ecology. We get this ecological understanding of human microbial entanglement, a kind of deep green on one reading, sense that the human is part of the world, and we need to think about our enveloping, our entanglement of the body into the wider environment. Okay, so there's an interesting alternative ontology emerging here that has been much celebrated by social theorists like Donna Haraway in particular. And alongside this is quite a radical different sociology, a radical different idea of the interactions between the elements in society. So the first of which is a story that's much more premised on mutualism, on symbiosis, on the sharing of genetic material, the sharing of resources, the need for interspecies collaboration over competition. And the second dimension to that is a radical decentering of anthropocentrism, a sense that humans are not separate from the world, they're not divorced from the world, that we need microbes to keep us human. And again, steps into a zeitgeist ecology around concerns around the Anthropocene, for example, and concerns about dysfunction and dysbiosis on different planetary scales. Much of this has come to focus recently in discussions about fermentation. Fermentation has become this very ripe metaphor for a strand of thinking in the environmental humanities that wants to take it as something beyond a metaphor, as a platform for a progressive political project. And this is just to take one example of a recent paper that featured in the journal Environmental Humanities, in which fermentation is being seen as a grounds here for a particular model of feminism. Fermentation here is being asked to do quite a lot as a metaphor, but as a manifesto foundation for a political project. Okay. So we can think here about an emerging sociology associated with the microbiome, in which the microbiome becomes metaphor, but becomes more than metaphor, it becomes a manifesto commitment to a particular way of resetting social relations and ecological relations. So that would be a second context I think it's worth thinking about. And the third context, which is very much related to the second, is how the microbiome resonates with this wider ecological zeitgeist that's concerned about planetary breakdown, that's concerned about dysfunction on a planetary scale around what I talk in the book as an idea of the probiotic turn. And the probiotic turn is a set of connected developments in which science is looking at the consequences of the overreach of modern ways of managing life, and a sense that many contemporary problems, whether that's around climate change, whether that's around antibiotic resistance, whether that's around the rise of infectious disease, are to do with Anthropocene blowback. So the excessive application of antibiotic ways of managing life have created these diseases from within the heart of the modern agricultural health machine, and that are now traveling widely across contemporary society. And so the project here is to use life to manage life, to try and selectively intervene into the circulation of energies within an ecological system to reset that system to deliver desired functions and services. So antibiotics here goes well beyond just the use of particular drugs to tackle particular pathogens, to describe more broadly a whole range of systematic efforts to rationalize the human body and the natural environment to optimize human health. And this leads to the simplification, the control, the smoothing, the acceleration of ecological systems. It leads to dramatic decreases in infectious disease. It has led to significant increases in life expectancy in many parts of the world, often by substituting organic processes with inorganic or synthetic alternatives. So using particular biochemical substitutes for processes and functions that would have been delivered by ecological agents. So if we think about this in the context of microbiome, we can think certainly about the rise of antibiotics, these wonder drugs that led to significant health breakthroughs in the latter half of the 20th century. We can think about the rolling out of that technology of pharmaceutical intervention into agriculture, into the wider countryside, but also into domestic spaces, the ways in which various forms of quite aggressive chemicals are now mainstreamed and normalized as part of a domestic hygiene ritual, whether that's around the use of antibacterial chemicals in kitchens, or the ways in which various chemicals are becoming mainstreamed into paints, into furniture, into household products, such that on the back of your pillowcase, you might find a label suggesting that this is antibacterial, or in a pot of paint here that's used for children's furniture, that it's sold as being antibacterial as well. So a kind of proliferation of these antibiotic chemicals into a whole array of mundane lifestyle components. And similarly in the food system and in the ways in which we think about the role of microbes in and outside of the human body, a way in which the Pasteurian project has been amplified and scaled up to sanitize food, to minimize the presence of microbes in a whole range of foodstuffs, to control and domesticate waste as it leaves the human body and moves out into the wider landscape. And there's obviously much to celebrate here, and I don't want to dismiss the gains that have been made from these antibiotic approaches, but there's a growing sense that there is an overreach here, that the application of those antibiotic techniques has gone too far, and we're now living through a nested collection of blowbacks that stem from the overreach of many of those ways of controlling microbial life. And for example, would be the story that I shared earlier about this idea that it's the missing microbes in the human body that lead to dysfunction, and particularly dysfunction in the ways in which the human immune system is trained over the life course to encounter particular microbes, that leads to this excessive overreaction to the presence of particular microbes that leads to the rise of inflammatory and autoimmune disease. It's a very complicated picture that goes on here, but the broad story you get, at least from the narratives of these books, is that this is an epidemic of absence. It's a blowback from the overreach of the use of antibiotics. Likewise, the growing concern about zoonotic disease and antimicrobial resistance suggests that many of these contemporary diseases, perhaps COVID, though we're yet to really establish the origins of COVID, come from within the modern ways in which agriculture operates, the ways in which various drug regimes are used to control microbial interactions in intensive agriculture that create these ideal disease conditions for new viruses to emerge. And then, of course, with globalization, they spread very quickly, such that, in the words of Rob Wallace, big farms make big flu. So this is a second example of blowback on a microbial scale. And then scaling out from beyond the microbiome, we can think about the ways in which ecologists, natural resource managers have for a much longer period of time been concerned about the ways in which the rationalization of the natural environment, whether that's around controlling fire, controlling disease outbreaks in forestry, the creation of monocultures in forest plantations, or the acceleration of drainage regimes in rivers, create these conditions for extreme events, create these conditions for blowback as the system loses its resilience, as it's able to tip into a much less hospitable condition for the life that flourishes within it. And right up in the, to the words of James Lovelock and other Earth system scientists, to the broader story about the Anthropocene, that this is a tip from the Holocene conditions into the Anthropocene as a consequence, if you like, of the blowback of human fossil fuel consumption, of the consequence of that acceleration, simplification, control, and substitution that's been at the heart of the modern antibiotic model. So the microbiome comes along at this moment as a particular set of responses, a particular way of thinking about ecological interactions, a way of thinking about dysbiosis, and as a way of addressing them. And in some ways, it's interesting in terms of particular, somewhat fetishized strains of bacteria that might be used to intervene into these systems. But for me, what's more interesting about the probiotic response is less, here's a particular strain that's going to tackle this particular problem, but the ways in which it captures a more systematic interest in modeling, monitoring, and intervening into ecological dynamics. So it characterizes a kind of new mode of biopolitics, a kind of ecologized mode of biopolitics, which is about intervening into the energies within an ecological system in order to restore or reset that system back into a desired stable state. So it's about using life to manage life, which is sometimes about these keystone species, these keystone microbes, but it's also more systematically thinking about how some forms of life are let live and some are let die in order to secure particular ecological conditions. And in the book, I trace this across a range of different scales. This is a diagram with an awful lot of text, and so I'm sorry, I don't know what size screen you're looking at this on. But broadly, the columns going from left to right trace this historical narrative from the antibiotic model through to a set of blowbacks into the probiotic alternative, and the rows shift from the micro to the macro. So we move out on scales, and we're particularly interested in this context with what's going on in those top three rows of the figure, in which there are a collected set of antibiotic interventions through the 20th century, broadly associated with the Pasteurian project, a collection of blowbacks to that, and this emerging set of probiotic alternatives, which are all based on new ways of thinking about the salutary role of microbes to restore desired systemic states, desired functions, to help deliver health on both the human scale, but also on the planetary scale. Okay, so those are three broad themes that I think is interesting to attend to from a sort of history of science perspective as to why we might be talking about microbes now, how might we understand the microbial moment in the context of 20th, 21st century science and the relationship between science and politics. So why does this matter? What's of interest here? What's at stake in the microbiome moment, particularly for this project? So it's clear, therefore, that we need to think about microbiome science and probiotic management as social practices, as all scientific practices are. They are shaped by social norms around race and gender, nature and progress, etc. The microbiome doesn't escape this, although we might like it, although some social theorists might like it. It's still a social practice. And so we need to think carefully about the social dimensions of microbiome science and how they might come to shape the nature of the science that's done, but also the applications of that science into forms of healthcare. So just to give an example, we see some quite eclectic versions of microbiome science coming to the fore. Just to pick on one individual here, Jeff Leach, who runs is quite connected to the human microbiome research community, but for whom the issue is very much about the problems of modern life, and in which the solution to various forms of epidemic absence is a return to forms of human microbial interaction associated with, I guess, there's lots of loaded terms here, but a sort of neo-primitivism, like a kind of valorization of traditional human microbiome interactions, which for Leach are found in the lifestyles of the Hadza. Here, he's drinking water in a part of Tanzania to simulate human microbial exposures associated with the life of Hadza people. And there's lots that people have said about Leach, and they're kind of the ways in which this is entangled with ideas about race, about progress, about colonialism. And there's interesting work to be done there. But more broadly in the book, I try and map out a kind of color-coded typology of the different ways in which microbiome science is shaping certain political interventions. So although there is a common ontology here, it lends itself to quite a wide array of different political projects that are to do with, the bottom there, the kind of political economy of how microbiome science is funded and who benefits from it, from the left, from the right, there's a kind of anarchist version of the microbiome. We move up from the bottom. There's a very cosmopolitan version of the microbiome versus a fairly racist, fascist version of microbiome science, or at least the kind of rewilding science that it comes to underpin it. Above that, there is a very heterogeneous relationships with technology. So for some, the microbiome is best revealed through next generation sequencing and machine learning algorithms. For others, it's about returning to a primitivism, a paleo version of the past. Some on the top there, for whom it's very much about human mastery over nature. There's a kind of eco-modernist light green version of the microbiome in which we can achieve our enlightenment destiny as the god species, the sort of Homo microbes god species, in which we will know, we will rationalize, we will personalize the microbiome in order to optimize human flourishing. For others, this is very much a story about folding ourselves back into ecology. It's about recognizing the problems of human mastery and moving towards a deeper green version of human microbial interactions. I can say more about that in the discussion if people are interested. Secondly, then, not only is there all sorts of politics going on in here, there's also a very clear sense in which this is still about selective interventions into which forms of life are allowed to live and which forms of life are made to die. This isn't a kind of laissez-faire celebration of the wonders of ecology. It is, in the words of Heather Paxson, a post-Pasteurian project. So it's a selective set of interventions to decide who are the good microbes and which are the bad microbes. How can we optimize the work done by the good microbes while still keeping the bad microbes at bay, let's say. It is, if you like, a kind of calculated program of decontrolling the modern project rather than a laissez-faire idea that this is about releasing the microbiome from human control. And so from an environmental ethics perspective, we need to think about which microbes are held to be useful and which microbes are let die because they are redundant or unknown or unoptimized. And certainly one of the strands of this has a kind of neoliberal focus on work. Let's say the microbes that work for us will have their futures secured. The microbes that are redundant or unhelpful can quite quickly be rationalized and made redundant in a system that's about optimizing the human microbiome for human health. And then thirdly, to circle back to some of the discussions to begin with, we need to think carefully about the epistemic alliances that we as social scientists make with microbiology and with microbiome science. So on the one hand, we're being offered a much more palatable vision of the world in this ecological story of symbiogenesis, which is about mutualism, which is about sharing, which is about collaboration, which is about shared flourishing. But we need to hold on to the concerns that have been true to the social studies of science for some time, about the ways in which that is inflected by particular social norms, particular ideas about how science is produced. And this is a concern that's been raised by Heather and Stefan again in a review that they did nearly 10 years ago now on the rise of the microbiome as they put it, a new model organism for thinking about grounding social and ecological projects. They look at Myra Hurd's work, and particularly how it was reviewed by Maureen O'Malley, who's a philosopher of microbiology, and raises, I guess, the classic caution that social studies of science has always had about thinking about science in context. And they suggest that by removing from the picture how the science was done, as well as the ongoing revisability and contestability of microbiological findings, we're left with the sense that there is a fount of straightforwardly produced and accepted knowledge from which we can drink. And that's harsh. I think there is nuance there, but there is, in as much as the biologists are now telling us stories that we like, we need to be careful about taking this at face value as a fount of knowledge that we could drink from, let's say, in their terms. Okay, so just to conclude then with 3 points of reflection that might be of interest for the HealthXCross project. And it's fair to say that I think all of these are already very much baked into the project. This isn't a kind of veiled criticism that these are things that the project isn't doing that I think it ought to do, but just to flag in some ways the significance of this project and the potential it has to address some of these questions. So the first of which, as will be clear in the focus on ethnographic tracing of laboratory science and the travel of microbial knowledge out of the laboratories into the clinic and elsewhere, is that we need to avoid the risk of scientism by placing microbiome science in its social, political, and economic context. Sort of science studies 101, if you like, but we shouldn't lose sight of that as we become enamored with the potential of microbiome science. Secondly, we need to recognize the great variety of human microbial relations. There are still all sorts of antagonistic human microbial relations that are premised on individualism and competition, and that can be explained by, in some ways, previous models of biology, alongside this, perhaps more relatively recent interest in symbiosis, in mutualism and exchange. And so there are myriad metaphors that one could take from the non-human world, and perhaps it's generative to pick an array of metaphors, but also to think carefully that there are many different ways in which the microbial could found the social. And I guess the third dimension, which is an area that I've been interested in and I think is also core to this project, is how making this new alliance with science could allow social scientists and natural scientists to collaborate. So the ways in which bringing an anthropological concern for social values, for power relations, for colonial histories, could help to guide the transformative potential of microbiology to develop, I guess, what's becoming known as this kind of post-critical approach, where we're both skeptical about the social context in which microbiome emerges, but we work collaboratively with microbiome scientists to steer this science to really harness its potential for improving health in the future. I will stop there. Thank you very much for your time, and be very happy to answer questions or to have a wider discussion about any of these themes that I've just raised.
So, Jamie, thank you very much. Thank you very much for this talk, all important, all true, and I could not agree more. Just let me one comment on your last remark. Yes, this project is also a collaborative project with microbiologists. And one of the ambition of this project is exactly to work with them in pursuing the microbiome development into a more transdisciplinary framework. And I'm happy that today among the participants, I can see some names of microbiologists, and I really believe this is the way to go and work together. So thank you, Jamie. As I said before, questions are at the end. So just mark your name if you have any questions. We prepared for the second speaker, who is Hannah Landecker, historian and sociologist of science, but really, as Jamie, a post-disciplinary scholar. Professor at University of California, Los Angeles, where she is cross-appointed between the Institute for Society and Genetics and the Sociology Department. She's author of various scientific publications, including the book Culturing Life, How Cells Became Technologies, and she's now working on a book entitled American Metabolism. So, Hannah, thank you very much for being here among us today, and I'll let you the floor. Mi potete tanto un po' muovere il vostro micro?
Yes, everybody should get up and stretch, I think.
Yes.
Hello, and thank you very much for inviting me to attend the opening of this exciting project. I am very much looking forward to how it's going to unfold over time. And I think that Jamie and I were thinking similarly, and I'm happy to say that there's going to be a lot of resonance between what I have to say and what he had to say. And my aim really was to maybe give some methodological and theoretical prompts to how you might be thinking about your empirical work going forward. So this talk really comes out of spending a lot of time embedded in the life sciences at UCLA. I spend half of my life in the division of life sciences, and I became aware a few years ago that people were beginning to really talk about boundaries in very interesting ways. And so I'm going to talk to you today about compartmentalization. Let's see how this advances. Yes. And I thought I would start a little bit like Jamie with this question of why we, if I can use the collective we as social scientists and humanists, might be interested in studying the microbiome. What's in it, so to speak, for the social scientist and the humanist? Rather conventionally, I think it is understood that the sciences and the technological developments that Jamie reviewed so nicely for us just now, that we understand that microbiome science is of enormous social, practical, and economic consequence, and that the ways that people are thinking about manipulating, commercializing, understanding microbes is going to be of enormous consequence, or is already of enormous consequence for food, agriculture, medicine, soil remediation, biological and social reproduction, and so on. And so it warrants us getting critically engaged, because it's too socially important to simply leave it to the microbiologists. So this is one sort of reason why we study the microbiome. And the second one that Jamie also addressed quite nicely is that there's a fascination element there, that the microbiome has a philosophical and cultural impact on how we think about self and other, boundaries of the body, cultural understandings, or representations of the human and more than human life and their relationships. Now, this might be good enough to get on with, that it is an interesting and provocative domain for concepts, socially important, and it's a provocative domain for concepts. That could be enough. We could go forward with that. But what I want to do in this talk really is to use the practical example of theory in the life sciences to ask whether it can open new empirical methodologies for social scientists in the joint project of understanding what is happening with and to the microbiome in an industrialized, anthropogenically modified, profoundly modified world. So can we, I would like to say we can, tap the conceptual work that's now underway in the life sciences. And this doesn't mean scientism, it means engaging with it as theory, now underway in the life sciences for theoretical renewal in the social sciences that is directly translatable into empirical questions that we should be asking of the microbiome. Can that novel empirical work speak directly back to the practical questions of microbiome science and how to live better in light of planetary crisis, One Health, dysbiosis? So I think very practically and very materially. So I'm going to give you an example in this talk of how that might work. So my aim is this sense of theory out of science that Sophia Ruth and Astrid Schrader coined as a term quite a long time ago for a renewed empirical alliance of social and biological methods. And the means by which I'm going to try to speak to this aim is to talk about compartmentalization. The first step, of course, is to review what it is, what I mean by that. You are probably wondering what that means. The second step is to ask what is the significance of compartmentalization and its perturbation in the health and functioning of the body in the Anthropocene in particular. Why is compartmentalization theory, and what it might suggest methodologically, even to those among us who don't speak a word of molecular biology. Okay, so let's start with to be able to see my notes here. There, I've got it. Yeah. Okay, so let's start with compartmentalization. When I talk about compartmentalization, you may have in your mind the kind of structure of the building we're living in, right? It has doors, it has rooms, it has cupboards, all kinds of things like that. But when biologists speak about compartmentalization in a biological space, it can be a cell, but it can be a tissue or a body or an ecology. They talk about the organization in space and time of different things by different organizational means. Put most simply, when you have sequences of chemical reactions, as all life is composed of, you often need to keep them apart from one another. Sometimes reactions are sequestered and kept apart from one another by an arrangement, say, of protein scaffolding or a clustering of enzymes that carry out the reaction. So this is chemical compartmentalization. If you only have the enzymes that do a conversion in one spot and not the other, you have already organized in space where that reaction happens. Sometimes, more intuitively, it's spatial compartmentalization. Say in a cell, you have a particular set of reactions that need a higher pH. So you have a little organelle, and inside that membrane is a space in which that reaction can only happen because that's where the pH is higher. So membranes are a common mode of spatial compartmentalization. That's the middle one. And then perhaps maybe least intuitive is temporal compartmentalization, that you simply have reactions going on in the same space, but organized by day and night. And you probably don't think about it, but when you're sleeping, very, very different genes are making proteins than when you are awake. You can think about it just very practically. You're not eating while you're asleep, or hopefully you're not eating while you're asleep. And so you don't need to be making a whole bunch of digestive enzymes when you're asleep. So those parts of you sleep too. So temporal compartmentalization is a big part of how biological processes are organized, again, at a range of scales, all of the scales that this project will study. Compartmentalization, if you include the temporal aspect of it, is a node of very interesting theorization right now in the life sciences. And I just give you this little quote from a piece called Why Life Oscillates, to think about why it is that we might see these different modes of chemical, spatial, and temporal compartmentalization across all kinds of life forms, across all kinds of spaces. So tuning and synchronization of rhythms saves energy. Temporal compartmentalization allows polar events to occur in the same space unit. There are polarities in the universe of our body, which cannot happen simultaneously. Systole and diastole, inspiration and expiration, work and relaxation, wakefulness and sleep, reductive and oxidative states cannot be performed efficiently at the same time and place. Temporal compartmentalization is probably the most efficient way to mediate between these polarities. And I was very struck in listening to a talk around the molecular details of some process like this. And the researcher was talking about futile cycles. That if you have the substrate that's going into a reaction and the product all present in the same space, you often have the undoing and the redoing of the process over and over again. And this is called a futile cycle. I sometimes think that my writing process is a futile cycle, but compartmentalization is used to explain many of these patterns. So you, no doubt, will be most familiar from whatever biology you engage with the idea of compartmentalization from looking at images of the eukaryotic cell here, which has evolved many membrane-bounded compartments that keep various parts and processes separate. But compartmentalization, I just want to underline, is not just about cupboards and boxes, right? It occurs at all of these scales, from the smallest scale of organelles or protein complexes, or even just gradients within these microscopic spaces, all the way up to communities, the community scale. So a very nice example of the way in which compartmentalization functions as explanatory theory within microbiome science itself is understanding of biofilms. So this is a very beautiful image, actually taken from a time-lapse film of a biofilm. And I chose it in part because it's from a lab at UC San Diego, which is one of the sites I think of this project. And so this laboratory used microfluidics and time-lapse microscopy to observe biofilms as they were growing. And biofilms, of course, are a problem for things like antibiotic resistance and chemical control, because it's really hard to get the bacteria in the middle of this biofilm. They're protected by this communal structure that they live in. The researchers discovered that when the biofilm reached a certain size, it began to oscillate in its growth. So it would grow, stop, grow, stop. So it was oscillating. And this paper theorizes that the oscillating growth is a manifestation of this metabolic codependence or metabolic compartmentalization, which is a relationship that is organized spatially within the biofilm. Sorry, I'm just trying to see my notes here. So it's organized spatially within the biofilm. The interior cells at the center of this biofilm produce food, basically, produce a metabolite that's necessary for the ones on the outside to divide and grow the biofilm further. And so this provides the ability of the inner cells, who are the source of this metabolite, to periodically slow the growth of the outer cells, which otherwise, if they just grew and grew and grew, would consume all of the available nutrients for the entire group. And so the cells on the interior would be starved if there wasn't this periodic halting. By periodically preventing the growth on the periphery, inner cells ensure that they have sufficient access to nutrients. And yet at the same time, by keeping the protected inner cells alive, the outer cells are ensuring a much higher chance of survival from external agents such as antibiotics. And so they're arguing that this harmony is not some sort of generosity on the part of the inner cells towards the outer cells, but it is this kind of codependent relationship that has this temporal and spatial organization within the structure of the biofilm. So that's a nice example of how the theory of compartmentalization, and including this temporal compartmentalization, is very important in explaining phenomena that are of core interest in the microbiome science that we see today. The intestinal wall, of course, is also an amazing example of this importance of spatial and temporal organization. It's both a barrier, but it has to be a zone of transduction. Things cross it all the time. It is, of course, the place that we now canonically think of microbiome interactions, and where a very large set of microbes live is in the human gut. And now we can look at this picture and see it as a structural compartmentalization, but this structure is itself incredibly interesting. So the wall of the human intestine is a single layer of epithelial cells. So it is known as an epithelial barrier, those little blue cells that you see there. And it's quite remarkable even just to think of the fact that it's a single layer of cells. It's very intricately folded with many projections called microvilli. And perhaps surprisingly, this epithelium lining the intestine is sometimes called the body's largest endocrine organ. Because about 1% of the cells have an endocrine function. This is these beautiful images here of these specialized cells that dot the epithelium. So about 1% of the cells in the gut epithelium have an endocrine function. They sense the nutrients that are moving through the alimentary canal, and they sense bacterial signals, and then they react to those cues and signals by secreting hormones and other cell signaling molecules that go on to affect satiety and appetite and nausea and many bodily assessments of the things like the volume of the food or the amino acid content of ingested food. So looking even more closely at one of these intestinal crypts, as they are called, we see that this single layer of epithelial cells is only one part of the physical structure of the intestinal boundary and its compartmentalization. And the boundary itself is compartmentalized, though not by a membrane. And I actually have to get up and talk you through this, I think. So in this image, you see this is the single line of epithelial cells. And you have these things called goblet cells, and they are actually synthesizing and secreting mucus. And if you look carefully at this diagram of the healthy, generalized, it could be human, and maybe a mouse model of the human of the gut. You'll notice that there actually are no bacteria in this darker zone. This is the first unit. The bacteria are all living up here in the loose mucus. The bacteria are digesting fiber and they're making short chain fatty acids. And these short chain fatty acids feed these cells. They are an energy source. Most of our bodily cells use glucose. But our intestinal cells, these epithelial cells, they're fed by the microbes on these chain fatty acids. The mucus is food for bacteria in turn. But this structuring, right? They are kept close. They are kept close by feeding, but they are kept apart. Actually, there are antimicrobial peptides in this part. So it is compartmentalized. It has this very precise structure to it. And it is the basis of keeping something very close but apart at the same time. In this diagram, it's a similar kind of thing, and perhaps in this project we can have some thinking about the jelly bean diagrams and the outside-in geographical depiction. But this diagram is very similar. It shows still this barrier of clear mucus and the bacteria and the loose mucus. But it is emphasizing this layer underneath epithelium called the lamina propria. And this is patrolled by immune cells. And these immune cells are constantly in conversation with the bacteria that are living again in this, this layer. I won't go into detail, but there's a great deal of crosstalk between the commensal bacteria living in the loose mucus layer and the immune system. So bacterial metabolites such as indole signal to the epithelial cells to enhance the tightness of the junctions between them. So this is one of my favorite terminologies in biology is the tight junction. They look like boxes, but of course the cells themselves maintain the tightness of the space between them in this layer. The bacteria do their stuff. And one of the things that they do is that they signal to these cells to tighten the joints between them. So in short, you just have a huge amount of conversation that is going into the construction of the boundary in the first place. There is, in addition, a temporal component to this relationship. So I'm bringing back here the temporal compartmentalization. The population of bacteria in the gut also have a circadian rhythm attuned to when their host is awake and moving and eating or asleep. So all of this signaling and feeding relationship that I've described also has a temporal compartmentalization with metabolic activities in the liver being directly cued in time by activity of microbes in the gut. And I've been talking a lot about the human, or what scientists have used mouse models to say about the human. But of course, if it's theory, it has to work in a very wide range of settings. And there are many, many other organisms, such as this corn species in Mexico that pursues this similar kind of barrier, keeping apart and together barrier. So this is a thick mucus layer around aerial roots. And this mucus layer around the aerial roots of the corn is home to bacteria that are specifically nitrogen fixing. And these plants live in nitrogen poor soil, and the nitrogen fixing bacteria therefore fix nitrogen from the air, supplying the plant with a much needed essential element that it can't always get from the soil. So in short, in this first part of the talk, what we learn from the general theory of compartmentalization and the specific example of the gut wall is that togetherness or codependency is paradoxically a constant work of separated togetherness. Rather than being an autonomous being that converts the world into itself, the very way that we are beings with a distinct outline that maintains integrity over time is to be in constant dialogue and interlocution with these other entities. So gut integrity, I've just told you, is a continuous process of interlocution between ingested food, microbes, bodily cells, and of course, other things in the abiotic environment. Light is in there. The light and dark cycle is in there in participating in this relationship. Now, I want to emphasize that it really is not that humans and microbes dissolve into one big undifferentiated blob. And I was thinking about a picture of the human in the office chair from the Micropia thing that Jamie put up there. It's so wrong, right? That it depicts the human as an undifferentiated mass of microbes. And what compartmentalization would tell you is that actually it's a highly, highly organized choreography. It's not that you go down one scale and we are composed of our microbes. It is a set of highly organized sets of boundedness and the maintenance of that boundedness. Okay, so that was very abstract, right? Very much in the realm of concepts. Why can I sit up here and show you these jelly bean diagrams and tell you that this is how it works and all of these things? Well, it is because compartmentalization, integrity, dynamic boundedness have become an object of concern and inquiry and investigation exactly because of its breaching. And it's breaching under these conditions that we see again in Jamie's talk as a contrast between the non-industrialized and the industrialized. So this utopic view of compartments arises from a dystopic view of their degradation. An example for you of this worry, and one of the reasons why something like the tightness of tight junctions becomes a focus of work, comes from studies showing the paradoxical situation in which humans are becoming allergic to their food. So observing, for example, that almost all babies tested in a study in 2018 in Europe had antibiotics reactive to milk and eggs. While the rise in general allergy to the world is also manifesting as asthma, ectopic dermatitis, inflammatory bowel disease, and non-alcoholic fatty liver disease is one of the ways in which the integrity of these boundaries and their maintenance is a source of science and a source of concern. So what does breach these sort of relationships at the epithelium. And as I've carefully described, you might think of the epithelium as just the skin, but the entire lining of the gut is also an epithelial surface. And in fact, you could reorganize our whole sense of organs by looking at epithelial surfaces through the body. So things like surfactants, enzymes, emulsifiers, cigarette smoke, particulate matter, diesel exhaust, ozone, nanoparticles, microplastics, all of these in scientific studies have been shown to damage the epithelial barrier. But more important than damaging the barrier is intervening in the dynamic process of its maintenance. So the barrier is not a thing, it is a process. And when you put things like emulsifiers and diesel exhaust into the process, then the process of the maintenance of its integrity becomes compromised. A large number of these substances are encountered by humans as a consequence of industrialization, urbanization, and modernization. And in particular, which I think is interesting because it's not something that we pay so much empirical attention to, detergents used in laundry, dishwashing, and household cleaning agents have been shown to be directly toxic for the epithelial barrier. So this is from the same review talking about this epithelial barrier hypothesis and sort of summarizing the data from available studies. And many of them are from, say, employees in the detergent industry, individuals working as domestic cleaners, but also people exposed to these substances. We see this all over biology today. Again, this is not just a pile of empirical studies. It is becoming a theoretical rubric by which to understand different phenomena. Air pollution compromises epithelial integrity at the lung surface, which is linked to COVID-19 severity, as in this study of Northern Italy. And gut integrity, or the loss of gut integrity, becomes a better predictor of death than chronological age in model organisms such as Drosophila. So in this study, regardless of chronological age, intestinal barrier dysfunction predicted impending death in individual flies. One form of compartmentalization is linked to another. Shift work, which makes people eat and be awake at cross purposes to circadian rhythms of gene expression and microbiome function, results in physical compromise of the gut barrier. And again, also shift work and COVID-19 susceptibility have been linked through this mechanism. Indeed, the International Association for Research on Cancer has declared chronic exposure to night shift work a probable carcinogen, and light at night has been classified as an endocrine disruptor. So we see this entification, reification of things that are profoundly socially organized. Work, the scheduling of work, the lighting of cities. These things are participants in the maintenance and rupture of these boundaries. So let's move on to the question of why this is theory and not just findings, not just empirical findings. It is indeed more than a pile of experimental results. And I can report to you that there are interesting ways in which compartmentalization is appearing in systematic attempts to systematically rethink the organization of biological disciplines, the topic, but also to systematically rethink what it is to say that an organism is healthy or not healthy. So the authors of this recent review, for example, Hallmarks of Health, explicitly set out compartmentalization, it's in the red at the top there, as one of the fundamental categorical means by which to distinguish between what is health and what is disease. And they write, life would not exist without organism intrinsic barriers that allow delimitation, electrophysiological and chemical gradients, yet still allow gas permeation, signaling across compartments, replenishment, and detoxification. So they cite the integrity not just of the gut or the skin, but also the integrity of organelle membranes, such as mitochondrial membranes, where the electrophysical gradient is the basis of the generation of cellular energy as a key marker of health and disease. If a theory is robust, it should allow prediction. Thus, we see in the recent announcement of the discovery of the world's largest bacterium, which to researchers' surprise illustrates the principle that compartmentalization reduces entropy. So even though the classic distinction between prokaryotes and eukaryotes is the organization of genetic material into a membrane-bounded nucleus, this prokaryote blurs that boundary by showing just this kind of compartmentalization. I love it that this single bacterium is the size of an eyelash. You're going to think that I'm taking you far too afield, but here we are suddenly from the world's largest bacterium to the question of herbicide-resistant weeds. But this is actually the thing, right? This is not just about the microbiome. It's not just about microbes. The research on microbes at the center of health and dysfunction is leading to this cross-cutting theorization. So let's visit herbicide resistance to see how it works out there. So this graph shows you very rapidly increasing herbicide resistance in weeds such as amaranth that are increasingly exposed to high levels of Roundup and other control agents. So if compartmentalization is a predictive theory, then we might be able to predict that it will play a role in this phenomenon. And indeed, researchers are quite surprised to see that there are these strategies of metabolic evasion on the part of weeds that are targeted by these glyphosate-based pesticides that are not classically mutagenic and mutational evolution, but strategies of metabolic evasion. So glyphosate, the active agent in Roundup, works by inhibiting a plant enzyme central to protein synthesis. And the plants are responding in multitudes of ways, but I wanted to bring your attention to the one that has the arrow to it here. This fourth mode is sequestration by the plant of the herbicide. They basically stick it onto their cell walls and put it into vacuoles. So they spatially compartmentalize the agent as it comes into the plant body, so it never gets to the enzyme. It is kept physically apart from the enzyme to modulate the effect of the herbicide. So indeed, all of these mechanisms of herbicide resistance might be seen as metabolic strategies in one way or another, but this strategy in particular shows that the plant responds to a ubiquitous environmental pressure and an environmentally selective pressure by organizing it. So it responds by organizing a toxin in space and time. Okay, so this brings us finally to the question of whether this theory, which I have presented to you as something which is not closed and final and certain, but a space of debate and contestation and interest and research. It's interesting in its own right, in its own sake, from a philosophical point of view, all of those things. But I want to go one step further and ask, does compartmentalization give you as empirical researchers a different way to parse your field sites and your data sets, or different frames in which to ask questions? And so I thought I would do that by giving you an example of how that might be. And let's return to this table that I showed you earlier and note that 5 substances in this table are enzymes. Now, while we're used to thinking of waste products or byproducts of industrial pollution as toxicants that damage the body or damage the microbiome, here we see something a bit different going on. Of course, synthetic chemical pollutants are important for understanding the industrialized microbiome and the organization or disorganization of its boundaries. But one thing I think that has really been empirically overlooked in this field is the way that humans have already captured and industrialized microbial metabolism and then put it back into the bodies and spaces of life. So enzymes, of course, are proteins. They are things that take one substance and convert it into another form. And over the 20th century, one of the ways in which microbes have been industrialized, and I don't mean that metaphorically at all, I mean inducted into the realm of industry and into manufacturing plants and all of those things, is to harvest these enzymes from fungi and bacteria and turn them to all the purposes that you see here. Detergents and cleaners, they're used in animal feed, in pharmaceuticals, in food and beverage. Fungal enzymes are used to make the centers of candies soft, all kinds of things from the most banal to the most powerful. Often microbial cells are immobilized on columns in order to continuously use the catalytic power of their enzymes to make things. And these enzymes from microbes are actually fundamental to the organization of the biotechnological economy, but also are a profound material shift in what parts of microbial life are in us and around us. So the metabolic power of enzymes are fundamental to all kinds of conversion processes across a range of commodities and industries, which will only increase with fermentation science, directed enzyme and evolution. All of these reasons why we are welcoming microbes as the new green future. Now, if we simultaneously pay attention to the role of temporal compartmentalization in this story, we see that the rise of industrial enzymes was part of a post-World War II manufacturing revolution towards what is called continuous process manufacturing. So here we see the plant scale use of mold amylase in the World War II period. And of course, one of the great examples of the harnessing of microbial enzymes is in the production of high fructose corn syrup. And so an example of an industry that underwent expansion through continuous process manufacturing in the post-war period through the power of enzymes to convert sugar and starch, interconvert sugar and starch, in this case, the enzyme glucose isomerase, which was capable of converting inexpensive cornstarch to high fructose corn syrup, utterly changed the sweetener industry in the 1960s and participated in driving the percentage of the population engaged in shift work towards its current levels of 20 to 25% of the workforce. So the food industry was transformed by the power of enzymes toward a different scale, but also towards a different temporality. This is just a nice image for you from the National Biscuit Co.'s new Chicago bakery, opened in 1952. And this is from Fortune magazine, which is celebrating Chicago industry by night, and writes that this is National Biscuit Co.'s biggest factory that will produce $55 million worth of cookies and crackers a year. And I'm quoting from the text in the magazine that's underneath this picture. Against the aluminum wall, abstract patterned with the reflection of fluorescent lights, stands a trough of dough ready for one of the plant's 12 600-foot-long band ovens. Each oven makes 15 million saltines from dusk to dawn. And every step of the National Biscuit Co.'s process is done by using these enzymes. So, as I said, one form of compartmentalization is linked to another. Shift work, which makes people eat and be awake at cross purposes to circadian rhythms. is a historically, culturally, socially formatted thing built out of the industrialization of microbial life. So it's not that we are coming to the microbiome and seeing it as damaged as by industrialization, but that it's already all together in there. And using the theory can open out these questions to things that we as social scientists might not think of as significant in the historical or the contemporary field. So seeing the enzyme, seeing the role of the enzyme in perhaps participating in these things which are under the general rubric of industrialization is a way to get to this opening question or premise. Can we tap the conceptual work to then ask these new kinds of empirical questions? So I just want to finish with a few methodological lessons or cues. I think that the industrialization of microbial metabolism itself, its processes, objects, and relations, is something that needs to be in the picture, as it were. The microbiome is not just a thing out there that we study, that we have hopes for, but it is in us, in our rhythms, in our biochemical milieu, in very profound ways. And as I said, this idea, fermentation is not just a nice thing for feminist hopefulness. It is being seen as, you know, the hope of the green bioeconomy and the ramping up of the bioinformatic basis of understanding which processes to scale up and work with is underway. So this is something I think that we as social scientists should be paying attention to and have the specific skills to understand. The history of shift work is not something I think that most microbiologists know a lot about. So this, I've already said it, historical reflexivity about the basis of the conceptual frames, I think is very, very important. How do we come to our positioning in this field of concepts and things? How have the concepts and things already made us what we are, formatted the problem that's in the dish? Be material about materialism. So where I think a lot about Marx, who used metabolic theory as a resource for political theory, and where Marx might have been fixated himself on fermentation and production. Today's theory, we could say, is being built on the matter of diabetes and irritable bowel disease. So being attentive to the substance of your theory, I think, is a methodological lesson. And finally, just my own personal work. Don't get too wrapped up in flows. Just because it flows doesn't make it good. We need boundaries. We need barriers. And it is an ethical question. So can we have a framework for viewing human action in relation to a biology that foregrounds a scientific and philosophical understanding of life as this organized process? Can we ask what allows people, societies, microbes, ecosystems, metabolic communities to maintain chemical, spatial, and temporal compartmentalization? Should integrity, the capacity to maintain, repair, and practice compartmental integrity, be a value to pursue across scales when we ask for concrete alternatives to current modes of doing things? And I've tried to give you examples for this. Shift work, light at night, as social justice. It's a biological thing, but it's a deeply social question. Are detergents and emulsifiers and these other banal, unnoticed, seemingly inert substances, are they physical agents of the modernity that people are speaking against when they say we need to practice decolonization? Nicole Redver's work, for example, on molecular decolonization, or Kyle Powis-White talking about communities having the capacity for collective continuance, I think has a lot of resonance with this idea that compartmentalization and dynamic boundaries. They are biological, but also simultaneously a social and an ethical formation. And that is where I'll end. And here are some of the scholars that I've depended upon in putting this together. Thank you very much.
Okay, so thank you. We received today 2 gifts. Thank you so much. We had an introductory talk from Jamie into the microbiome field, and his concluding remarks as smoothly introduced to Hannah Landecker talk, who has put us down into the materialities and substance, opening up this conceptual methodological space for transdisciplinary collaboration. So thank you so much. We now have our space for questions and debate. If anyone has any question, please write your name on the chat or prompt into the discussion. Here in the room, I already see 1 question, 2 questions, 3. That's very good. So please just, maybe you can come here. You can pose your questions. Oh, can you please come here? Thank you.
I think that we can't hear you unless you speak.
Okay, è per questo che...
This is Davo. My question is, maybe you can answer.
Sorry, can you?
I was wondering if we know which microbes gut microbiome are those related, specific diseases in particular mental disease. And so how, if so how we can reshape any microbiome or filter with that microbes.
So I think the question is, do we know which species or which kind of microbes in particular may participate the most robustly in maintaining a healthy gut boundary and those kinds of things. And I think that this is one of the things that Roberta and her team are really studying, is that the identification of who's in there, as the scientists say, is very much based on techniques that maybe are not paying so much attention to specific species. They are mapping the genes and to some extent mapping the metabolites and asking whether clusters of gene activity are more associated with scenarios in which you see a state of health versus a dysbiotic state. But it's, this is a very open and emerging field, and I think that many people are experimenting with the kind of probiotics that Jamie was talking about, to say, well, if you feed this into the participant community, then you seem to have a better outcome than if you feed that. So it's not at the level of this species or that species. It's very experimental, and also these questions of how much it has to do with community structure and the relationship between the epithelial cells and the microbes. So there's no good answer to your question, which is, well, how should we treat
Yeah,
that? But there are a lot of indications that various health proponents will tell you that you have to feed the microbes in the gut, that you have to feed them fiber, that perhaps they're eating things that have emulsifiers in them. Emulsifiers are basically detergents. So if you want a healthy biofilm layer on the surface of the colon, you probably shouldn't fill your gut with things that I mean, what is an emulsifier? It's soil and water mixed together in a undifferentiated way. So this is what I'm saying. Everything should not flow. The taking in emulsifiers is perhaps as important as which species live in there. So I think there's a great deal of hopefulness in this field for finding the things that will allow us to cultivate those boundaries in a good way. I don't know, Jamie, do you want to add to this answer?
Thanks, Hannah. I mean, I don't have specific species or even compositions of microbes. I mean, I think what's interesting around this is the temporalities that you raise, and particularly the idea of baselines and how one would index health to some place or moment in the past to have a reference. So, I mean, there's particularly kind of toxic relationships that you're diagnosing, but caught up in a lot of this is a certain sense that modern life is rubbish and it was better over there or back then. And I don't know whether that's a useful thing to open up in this wider discussion about time and its relationship to the microbiome.
So I just want to add right now something came to my mind. So for example, I have some mental issues, mental disease. We go to psychologist, psychiatrist, and he or she just recommend, okay, you have depression. So, but you say, I mean, based on microbiome science, you say, okay, mental is also related to microbiome. What's happening on your gut? So it's probably making you, I mean, affecting your mental health or physical health. So, but there's no relation with, you know what I mean? For example, because I found out some physical diseases, I mean, are related to mental diseases. So, like here, the diagnosis is, somehow, I mean, false. Because, for example, maybe specifically some diseases in colon, or like IBS is related to mental diseases. So, but here, the psychiatrist just prescribe antidepressant with these things. Not maybe the problem is just for if you fix the problem of your colon, so the problem will be solved. Yeah.
Here. Do you want to sit here? Or would you like? I'd rather stand if that's all right. I mean, I think that it's a great question, and again, it was something that Jamie was talking about, this idea of a gut-brain axis, and the sense that many of the drugs that we've developed for treating mental illnesses act on neurons. So they think that thought should in the neuron in the head and all of those things, but there are many connections between what's happening in the gut and happening in the brain. Unfortunately, I don't think if you did go down to the psychiatrist's office today that you would see that kind of emergent thing. But as social scientists, we often study communities of people who take it upon themselves to experiment and treat. And Jamie has done a lot of work with this kind of collectives of patients, maybe more with digestive diseases, but I think it's also happening in mental health of experimenting with probiotics and experimenting with different kinds dietary treatments that are pointed towards cultivating the microbiome for these issues. And it is also an intensive aspect of research. We had decades of autism research before people then began to say, well, what's the microbiome doing in autism or these things. So I think it is an emerging arena that will have a lot of things going on and that you all are going to study in the future.
Thank you. Thank you very much for this question and for the answers. And if I just can add something, you said, so the diagnosis I received now is false.
Yeah.
It's not necessarily false, but the mental state is part of this process of the dynamics of the organism. And so it's not false, but it's partial, probably. And why the microbiome research is so interesting is because it's bringing in a revolution in how we conceive health as these complex dynamics involving co-dependence, communication between cells, between microbes, between organs. So we are entering at this stage, and some the microbiologists usually say that we study who they are and what they are doing, referring to microbes. So there are the two sides. So it's important who they are in a way, and science is multiple, it's not just one. So there are some scientists looking more at who they are, but also more and more they try to integrate to the understanding of what they are doing. So looking also at the process, because you can also be Bruno, but depending on the context, you might behave as Sylvia, or, you know, it really depends. So it is a science of moving, and it's so exciting to chart what is happening. Thank you. So there were other questions down there. Yes.
So thank you very much to both of you for your event and inspiring talk. My question is maybe more just to Professor Lorimer, because this touched this point, the second point of the talk, but of course from the clinical view. So while I was pointing at the huge topic of the COVID emergence, I've read that it is less probable that diseases emerge from intensive farming industries than, for instance, from the wet markets, because of the biosecurity infrastructure that is in force, and also because of the our regional livestock and homogenized in history. And so, yeah, I was wondering what you think about this topic and if it's true that emerging is less probable in farming industries, or yeah, and or if there are possibilities of epidemic emergencies increasing exactly because of this biosecurity. Thank you very much.
I think that question was directed at me. Was it, Roberta? Obviously, I didn't pick up all of the what the question asked, but if it's broadly a question about whether modern intensive livestock agriculture has created the conditions for new diseases to emerge, particularly to do with the way in which they've reconfigured the microbiomes of the animals and their immune systems, I think the broad answer is yes. And quite well evidenced, particularly by virologists, as to the emergence of new strains of viruses that come out of factory farms and then spread through the vectors of globalization. I think in part to do with the use of regimes of antibiotic chemicals that are now structural or kind of infrastructural for the production of chicken and pigs, and to a lesser extent beef as well. I noticed that Fred Keck is in the audience. I don't know if I can put Fred on the spot, because his area of expertise, he may well not be available to talk, but there is certainly a great group of interdisciplinary social science going on around these hotspots of zoonotic disease and what might be done to both understand their geographies and causes and how to regulate them better.
Yes, thank you. I don't know if Fred is available, or you might want to add something about it, and especially the role of lab market. Maybe for Hannah, do you have something?
So I would just add a kind of reversal of the thinking in the sense that there is the question which I think I succeed to declare on about novel viruses, say, or novel bacterial pathogens being particularly well housed in industrial factory farming or those kind of things. But one can say, with the scientists who are thinking about the host rather than the pathogen, that the system of industrialized factory farming has changed the human body in a very profound way. And of course, it's only one component of the reason why we see a worldwide rise in adiposity and obesity and diabetes and non-alcoholic fatty liver syndrome and all of these things that raise the likelihood that the person who lands up with a serious illness or be hospitalized very human. And so you could say that the factory farming is not so much about whether it gives rise to this pathogen or that pathogen, so much as what are the ways in which these monocrops, these biocidal regimes, these productions of mass amounts of one kind of thing have changed the biochemical milieu that is disease transmission. And so I think one of the things that we should ask more is not so much where did the virus come from, but what made the susceptible body and made it susceptible in the way that it is. And we could ask as a thought experiment about the historical particularity of COVID, not as a new coronavirus, but as a very particular historical moment of the relationship between part of post-metabolic processes and post-metabolism. What has even tried to the historical biology that made play out there. And so you see that as, you know, factory farming is definitely implicated in it, that it gives you a kind of different framing for the class. Did you want to turn to an online question?
No, there is one question here in the room. If you can please. Maybe it's better if you, because from online they're saying, dicono che c'è l'eco, non riescono a sentire molto le domande. So it's better if you come here, please.
Yeah, no, no problem. I can stand. You don't need to get up.
No, they're saying it's better.
Okay. So I should sit here?
Yes.
Nice. So, so I actually have a question for each of you. So I was wondering about the first talk. So sorry, I should say that I'm a guest here. I'm a I'm a system immunologist and a theoretical biologist. I'm here on sabbatical, and one of the things I want to do on my sabbatical is, so because I'm an immunologist, also interact more with this question about how we talk about the microbiome and also how we talk about the immune system. And there were two things I noticed in your talk that maybe as an immunologist or theoretical biologist were missing for me. One is that on the one hand, you didn't really talk about the immune system, which, as we heard in the second talk, is really important for understanding how both the pathogenic and the non-pathogenic microbiome in our body lives. It's husbanding this microbiome, and it's actively doing that. And on the other hand, I'd like to I wonder what you think about the idea that it's not just the good microbes and the bad microbes. There's also a huge range of neutral microbes who could care less whether humans ever existed or not. And actually, if those microbes disappeared suddenly or were really influenced, that might be much more dangerous for us than anything we could do. Or just basically the idea that a lot of the biome doesn't care about humans and is not good or bad, but just there. And what does that mean for this whole discussion that we?
Thank you. I think that was a question for me, was it? I mean, those are
yes.
important points. And yes, certainly there is a tendency to see everything through the eyes of the human host here and to ignore our irrelevance to microbes, in many places. And the immune system as well. It's something I've written a little bit about elsewhere, but not as much as others. And the ways in which, in some ways, our thinking about immunity has been reconfigured with the emergence of the microbiome and this move to a much more ecological understanding of immunity, if you like, than the kind of Marshall model of self-other that's going on. And again, this is not something that I can claim to have come up with, but I think as a wider way in which our theories of immunity have come to adapt to the kind of Anthropocene zeitgeist is something that it would be fun to think about without necessarily folding it into this flow type ontology that Hannah's right to caution about. I think of kind of Roberto Esposito's ways of thinking about immunity and hospitality, which are about, in some ways, securing the host in its hospitable relations with others as a useful way to go. But there's a big literature there that we could talk about at length, I'm sure.
I'll just say one thing, that I just organized this meeting of people studying non-human immune systems just this last summer in Dresden. And one of the interesting things there is that there was a lot of people talking about how the immune system is maybe mostly there to make sure you have the right biome, not so much to protect you from microbes. And other animals do very strange things with microbes that we don't even begin to imagine. So my second question to you, or maybe I don't know. So I'm a contrarian. Okay, I can't help myself. So when you said that there's a rise in diabetes and in all kinds of gut issues and things like that, it made me think of two things. One is, what if rather than the change in microbes, actually the first thought I had is, okay, but there's also a huge rise in lack of starvation. And that brings me to my second point, is many people alive in this room, included, but definitely in the world, in most other ages would never be alive. Because the main thing we've done is we've stopped people under the age of 5 from dying. And so what if, again, not just the fact that we eat new foods, but the fact that there's lots of kinds of people who usually would be dead and never become adults are around and having kids. Okay, so now we have all these people whose biologies are maybe not as adaptive, but can be alive today. They can be alive today. And so maybe that's part of why we see these diseases. Not so much because of the influence of outside things, but because we are now much more diverse than we ever were. And some of that diversity is people who have not such adaptable phenotypes. And of course, I think they still deserve to be alive. And I'm happy they're here, but they give rise to diseases that we haven't seen before.
How to answer that? So the first thing is that one of the things that I think is being produced by the social, political, economic, and therefore scientific concern with diabetes and obesity and that range of chronic metabolic disorders, regardless of its actual cause, is a transformation of the sciences that you yourself participate in. And so, you know, the rise in, for example, thinking about chronic inflammation and the spectrum, the biological spectrum that goes from acute inflammation of swelling and reddening and heat to this emergent and powerful sense that this shifted metabolic state of lots of adipose tissue and these kinds of signals gives rise to a chronic inflammatory state. And so that, whether or not and whatever the cause is and the really real of the epidemic in chronic diseases, working on that as a problem has changed the nature of biological thinking and has changed the nature of biological thought. And so it isn't my role as a historian and a philosopher and a social scientist to say, okay, the cause of obesity in the world is X or Y, and here are the arguments for it, and here are the arguments against it. So that's one kind of answer, and you might find that to be evasive, but it's not evasive. It's just that what I care about is the shape of thought and what drives it, and then what it does. And, you know, we see the rise of this ill-named but interesting field of immunometabolism, or the realization that epigenetic processes are not just the kind of decoration of genes, but in the mitochondria, you have the production of all of these metabolites that do all of that. And so there's this kind of rise in the agency of these entities that were before seen as storage depots or power plants. And so it's fascinating. This transformation in thought is in large part driven by, you know, the NIH dumping money into chronic metabolic diseases. So this changes the way that we think, we theorize, we do. And that to me is what's important there. But then I'm going to go back on my own thing and say, well, no, it's not just that we're living longer. It's not just that somehow, you know, there's this great cosmic trade-off between living long enough to get old and being sick and not living long enough to get old. It is clear to me, and I think to many other people, that the things that we put into the atmosphere and the water and the soil and the chickens and our babies in the 20th century, in the name of health and vitality, those things are biologically powerful agents, and they are participating in our biology in ways that we do not fully comprehend, and we should, because many of those things are actually quite straightforwardly actionable. We actually do not need to have PFAS in our dental floss, right? If PFAS is a profound interrupter of these substances. So there is this role there for saying, you know, whatever it is that the cause of these things is, we know that low dose arsenic is a diabetogen. We know that various kinds of organization of day and night, which impacts the lower socioeconomic parts of the world much more deeply, that those light and dark cycles, which are totally structured by social action, are through the circadian metabolic immune link, they are extremely consequential, but we don't act on them as life and death matters. We act on them as like, who will do that job for that pay? So that's the second answer, that there is a material thing there that we do need to understand together, not just as life scientists, but as life scientists and social scientists. So that's the second.
I like the first one.
He said he liked the first one more.
We can ask two more questions. Also one question.
But we should talk more because I think some of the examples. Well, I love the mitochondria.
How it works. I ask Bing to jump in and ask the question directly, please.
Hi, good afternoon by now. Thank you, Hannah and Jamie, for always very inspiring talks. And my question is a bit oriented towards both of you, and it's a more general question, because I've listened with great attention to your talks, and you make use of a lot of existing research to build your argument, which we all do, actually. But you make a distinction between the empirical research as well as the theorization, and trying to come to a proposed meta-theorization that would work across the disciplines. Therefore, the fact that there has been a lot of research, empirical research before, as well as social theorization before, makes that possible. So it's transdisciplinarity rooted in disciplines. But then I'm wondering how to go about in aspects of under-researched topics where you have very little empirical research or little social theorization. And that connects then to the question of Jamie Lorimer on how we can do the last bit of your list, working together with the scientists, for instance, to try to affect it and be part of giving it some direction, and while still being critical from a social science perspective, thinking about the thoughts, but then also really shaping the very science of it. So yeah, and I'm asking that question in relation because now we are often talking about human microbiomes and the processes, but most of that is based on Euro-American populations, more than 80%. So, and very little is done in non-Euro-American settings, which makes, and we know that there is a potential for huge variation in these gut-human environment interactions across the world. So that could perhaps subvert a lot of the things we are doing. It's just, yeah, how would you go about doing transdisciplinary research on things that are very little researched? That's actually the concluding broad question.
Do you want to go, Hannah?
What?
I'm just wondering if Hannah wants to go first.
Yeah, I think the microphone is off. Hannah, yeah, the microphone is switched off.
Okay.
Yeah, okay.
So Jamie, why don't you go first? Because I just talked a lot. You have a say.
Yeah, it's a great question, Vim, and thank you for asking it. I mean, I guess both Hannah and I, by virtue of what we study, which is scientists doing science, are somewhat hostage to the science that the scientists do. And although we might want to steer them towards working on other people or other questions, we're obviously constrained by the political economy that funds health sciences, which, as we know, tends to focus on a subset of diseases that are of interest to a subset of the global population. So those are big and important questions that microbiome science faces alongside other groups. I think there's definitely scope to steer the direction of that and to work with perhaps more maverick parts of the microbiome science community who are interested in populations and phenomena in other places. I mean, I think to circle back to the theme that I raised earlier about the ways in which other places and other populations are sometimes held to offer the reference database for pre-modern, pre-industrial life does figure, and it's quite a problematic geography and quite a problematic history to do with that. But that's a way in which some people in some places have become folded into these databases, albeit they come to serve a particular function in the imaginations of what went wrong and how we might get back to it. So you move into that space with a degree of trepidation, I think.
Yes, I think I would just second that, but I think that there is also perhaps when you look at arenas of research that might not be labeled microbiome, for example, all of the work in antimicrobial resistance, the Antimicrobials in Society project at the London School of Tropical Medicine and Hygiene that both Jamie and I think, are advisors on. I think that actually presents an amazing model for empirical work all around the globe, and asking not some kind of universal question that you then drip over the globe and it will run evenly over all of its surfaces, but to say what is the question in this site that might bear on the science of the microbiome, or in their case, antimicrobial questions of antimicrobial use and antimicrobial resistance. So I do think that there are some models out there. I think that there is also increasing interest in the scientific community in community engagement of various kinds, and I think one of the sites in this project in Hawaii, if I understand it correctly, is explicitly working with scientists who want to ask questions that are specific to where they are and who they are. And so there is some of this kind of local biologies, I would say, speaking back in different places. And of course, collaboration is what we should do. So collaboration is the answer as well.
Okay, thank you very much.
Thank you very much. I mean, the event should have ended at 12:30, but there are 2 more questions, so I'm happy to welcome the questions if you can keep short. Maybe we should take both questions together and then we have to. Thank you. So please come here.
Can you hear me? Okay. Thank you so much for the talks, both of you. Very interesting. I actually have a question for the team of HealthXCross. And I'm a student of a master's degree here at Campus 3, Environmental Humanities, and I'm writing a thesis about migration and climate change. And I was wondering if you are considering to include, or if you already are planning to study how climate variability and climate change influence, together with human migration specifically, influence or impact health from microbiome perspective. I don't know if it's in the same, like in a correct way, but and if you're considering to do these, how are you planning to integrate these topics in your research?
Yeah, my question was inspired by Professor Lorman's talk, but actually I think it also goes hand in hand with the suggestions Professor Lambert gave. I was wondering, regards to the advice to recognize the variety of human and microbial relations, and in particular to the selective interventions that Professor Lorman talked about, to decide who and what gets to live and to die. I was wondering, how to, as an anthropologist, how to find the balance between, the limits of, on the one hand, of biopolitical controls of microbes, without, on the other hand, of course, neglecting the risks of multi-species relations. I'm thinking in particular about immune depression that may be induced by chemotherapy, for example. And so how to, how and what tools to choose methodologically in positioning ourselves as ethnographers between different players. So, there may be doctors on the one hand, describing chemotherapy with a very scientific approach to, with a particular aim of deciding who's in and who's out, and then more integrative alternative medicine approaches that, that study the complexity of our immune systems. And I think my question is very much tackling with the problem of how to avoid just using microbes for metaphorical purposes, and pick and choose the stories and the practices we need to tell our stories well, but to actually really take seriously our interlocutors without ignoring the political implications that they have, because I think that's how I interpret the work of anthropology. To be working with the microbe.
Can I just, sorry.
Thank you for these questions. This is a fantastic question to wrap up the debate. And your questions, we will take into account. This for all the team we are inspired with.
Jamie, do you want to speak to that last question?
I can try. It's obviously quite hard to generalize across the range of different situations that we might find ourselves in terms of understanding appropriate relations with the scientists who have some traction over controlling microbial life. I think it's also fair to say there's still a high degree of ignorance in this space in terms of actually knowing what does what, let alone ability to discriminate in terms of killing some off or enabling others to live. So we should be cautious about assuming that there is that much control going on, or that we need to be too alarmed about some coming microbial apocalypse that's going to be let loose by a probiotic management regime. I guess my concern is a tendency that's been true for a while to try and reduce a function to a thing that can be put in a pill, if you like, and the ways in which that might constrain microbial flourishing, that you can reduce it to a single agent that could then be universalized and patented, that they may well have consequences for microbial diversity, which we see in some of the ways in which the fecal microbiota transplant might be turned into a more readily commodifiable crapsule, if you like, as they call it, that might lead to a loss of some elements of microbial diversity.
I guess I would say that you need to find the contrarians in the scientific community. The scientific community is not a monolith. They have their own discussions and their own arguments and their own contestations about the very things that we would want to keep open to contestation. So if you want to think about it in terms of classic anthropological methodology, this is what George Marcus used to call para-ethnography, that you are an observer among other expert observers with their own open questions. And so, I think just as a methodological prompt, finding interlocutors who are willing to argue with you and each other, and to take questions as open rather than closed, is just a good starting place. And there's a very kind of fervent field of consideration and debate and entrepreneurialism and competition and creativity and all of these things. So find the places where it doesn't make sense, or people are at odds, and allow that to be a place where you can enter into that conversation.
So thanks for the questions, for the answers. Thanks for being here with us, being with us all this time. And I thanks also the technical and administrative assistants for making this possible. And so lots of food for thought. Thank you very much. And I hope you will join also for other events. We will continue this adventure, and see you soon. Thank you very much.